In this video, we're going to talk about the chemical structure of DNA, or deoxyribonucleic acid. The three-dimensional structure of DNA puzzled scientists for years, but in 1953, James Watson and Francis Crick announced the iconic double helix shape of the DNA molecule. But it was an image of the molecule taken by researcher and crystallographer Rosalind Franklin that was key to deciphering its structure. And the image was basically removed from Franklin's desk drawer by Maurice Wilkins and given to Watson and Crick, who were then able to finally develop a full understanding of the three-dimensional structure of the molecule based on Franklin's image. But Watson, Crick, and Wilkins received the Nobel Prize in Physiology or Medicine in 1962 for their work, but unfortunately Franklin died of ovarian cancer in 1958, 4 years after Watson, Crick, and Wilkins received the Nobel Prize. The classic DNA double helix is the broad view of DNA. Here we're going to take a closer look at the components of the molecule, the chemical structure of it. DNA is made up of subunits called nucleotides. A nucleotide consists of 3 main components. There's a 5-carbon sugar, and note that this sugar in DNA is deoxyribose, and this pentagon shape has the 5 carbons that it's talking about. There is also a base and 1 or more phosphate groups. The 5-carbon sugar and the phosphate groups form the backbone of the molecule. And note that the phosphate group has 2 negative charges on its oxygen atoms, and that gives DNA an overall negative charge. Recall that nucleotides of DNA normally contain one of 4 kinds of bases, denoted as A for adenine, T for thymine, G for guanine, and C for cytosine. So 2 of these bases are double ring structures known as purines, and they are adenine and guanine. The other 2 are single ring structures known as pyrimidines, and they are thymine and cytosine. The combination of a sugar and a base is a nucleoside. If we add 1 or more phosphate groups to the nucleoside, it becomes a nucleotide. For specific naming of nucleotides, we can look at the number of phosphate groups. If there is 1 phosphate group, the nucleotide can be called a nucleoside monophosphate. Remember, mono means 1. If there are 2 phosphate groups, it is a nucleoside diphosphate, because di means 2. And if there are 3 phosphate groups, it's a nucleoside triphosphate, and tri means 3. So let's just refresh our memory of the structure of a nucleotide. There is the phosphate group, the deoxyribose sugar, and the base. So let's add the molecule of adenine as the base of this nucleotide. Also remember that DNA is a nucleic acid, and that nucleic acids are polymers of nucleotides, which just means that the DNA molecule is made up of a chain of nucleotides. Nucleotides are linked by phosphodiester bonds. So in a nucleic acid, which is a polymer of nucleotides, each nucleotide sugar is linked to the phosphate group of the neighboring nucleotide. So this phosphodiester bond is this carbon-oxygen-phosphorus-oxygen-carbon linkage that connects the 2 nucleotides. So you have the carbon here, oxygen, phosphorus, and oxygen that forms the phosphodiester bond. The strand of DNA has polarity, which means that one end differs from the other. At the top, of the figure, we have this free 5' phosphate group, and at the bottom we have a free 3' hydroxyl group. By convention, if a DNA sequence doesn't specify the 5' or 3' end, the end at the left or the top is the 5' end. The phosphodiester bond is stable, and it can withstand changes in pH and temperature that might break weaker bonds. So if we were to read this particular strand of DNA, we typically read it in the 5' to 3' direction. So recall that the bases here are adenine, guanine, cytosine, and thymine. So to read this sequence, we would say that it is A, G, C, T. And we often put this indicator here of 5' attached to the sequence, and then at the end it's attached, and we say 3' at the end to give an indication of the direction. Watson and Crick were able to determine the structure of DNA by considering earlier physical models that indicated DNA was a long molecule, as well as base pairing rules that were established by Erwin Schargraf, and that important X-ray diffraction study by Rosalind Franklin that implied that the DNA molecule formed a helix with a repeating structure. So in a space-filling model or diagram of the DNA molecule, there are two strands of DNA wrapped around each other to form a helix coiling to the right. The sugar phosphate backbones wind around the outside of the molecule and the bases point inward. The outside contours form an uneven pair of grooves called the major groove and the minor groove. And it's important to note that the two strands are anti-parallel, which means that they run in opposite directions. In this ribbon model, it shows the base pairing between the strands. The bases point inwards, and base A only pairs with T and base C only pairs with G. The bases A and T and the bases G and C are said to be complementary. The bases pair via hydrogen bonds. So the specificity of base pairing is due to different hydrogen bonds that form between adenine and thymine that are held together by 2 hydrogen bonds, and between cytosine and guanine, which are held together by 3 hydrogen bonds. These hydrogen bonds are formed when an electronegative oxygen or nitrogen atom in one of the bases shares a proton with another electronegative atom in the base across the way. Because of the complementary base pairing, the total number of A bases equals the total number of T bases, and similarly, the total number of G bases equals that of the number of C bases. And these equalities are often written in terms of percentage of each base in the double-stranded DNA. But the percentage of A's always equals the percentage of T's, and the percentage of G's always equals the percentage of C's.